Hi, this is Xingfeng Shao from University of Southern California, and I have nothing to declare. In our previous study, we proposed a diffusion prepared Picasso technique to non-invasively measure BBB water exchange rate, Kw, which has shown good test-retest reliability. And the measure Kw can be sensitive to BBB dysfunction and the early stage of disease progression due to its smaller molecular size and special transport mechanisms. The DP Picasso technique has been shared with other research institutes that has successfully detected BBB dysfunction in Alzheimer's disease, small vessel disease, sleep disorders, and schizophrenia, and other diseases as well. Our technique has also been validated in animal stroke model and independently implemented on GE and Philips scanners. However, the diffusion preparation module leads to half signal loss, so the Kw map have very limited number of slices with moderate slice thickness. And the PSW, the permeability surface product, cannot be estimated due to lack of venous compartment in the modeling, and the simultaneous mapping Kw and PSW is desirable since they provide comprehensive information of the BBB function. So the goal of this study is to improve the SNR so we can obtain high-resolution maps with comprehensive information about water exchange kinetics, and which will further help to obtain individual CBF, Kw, and PSW maps. In order to achieve these goals, we developed a new pulse sequence, implemented 3-compartment modeling algorithm, and proposed a simple yet robust algorithm for individual Kw mapping. And here's a diagram of pulse sequence with the proposed motion-compensated diffusion weighted scheme. Since we only need a very small diffusion weighting to separate the intravascular and extravascular compartments of the ASL signal, we redesigned the crusher gradients of the grace refocusing pulses for diffusion encoding and motion compensation. However, we still notice the residual artifacts since very subtle residual motion can violate the CPMG condition of the grace readout. To improve the efficacy of motion compensation, we designed a non-selective adiabatic refocusing pulse, which successfully improved image quality, and the temporal SNR of the diffusion-weighted ASL image was 4.2, which was threefold higher compared to a previous DP Picasso technique. And MRI experiments were conducted on 3 Tesla Siemens Prisma scanner, and we collected data from 11 subjects. So here shows the perfusion images without diffusion weighting and with diffusion weighting. While the capillary signal was numbed by the diffusion gradients, the difference image represents the vascular signal. The vascular fraction is high and short post-labeling delay, and we utilize a multi-PLD acquisition to capture the dynamics of the water exchange. Vascular friction will decrease and longer post-labeling delays when label signal exchange into tissue. With a sufficient long post-labeling delay, non-exchanged spins will be drained into the venous compartment, and we need a venous compartment in the modeling for more accurate quantification of water exchange kinetics. ASL signal can be expressed as a convolution of the arterial input function and residual functions, and according to the SPA model, residual function of the vascular compartment can be expressed as in 2 stages, while the spins in the exchangeable capillary compartment signal decays with 2 time constants, the water exchange rate Kw and the longitudinal relaxation time of arterial blood R1A, and when the spins reach the non-exchanging venous compartment after capillary transit time tau c, signal will only decay with R1A, but not with Kw. So here shows simulated residual functions and corresponding ASL signals. The black and red curves in the acquisition window represent the perfusion signal without and with diffusion weighting. And we apply voxel-wise 3-compartment SPA modeling by solving this optimization problem. And here are the perfusion signal with fitted results from 11 subjects in green matter and white matter respectively. And for each region, we can calculate the CBF, VC, the capillary volume, Kw, and tissue T1 from those 5 PLD ASL signal without with diffusion weighting. And we can further derive PSW and the tau c. So here shows the fitted CBF, VC, and Kw maps, and with derived maps for capillary transit time tau c and PSW. So based on the 3-compartment modeling results, especially the capillary transit time tau c, we know PLD of 2.5 or 2.8 seconds are sufficient for complete water exchange in majority of the voxels, so we can estimate the total extraction ratio and calculate the PSW according to the indicator diffusion theory. So also based on the 3-compartment SPA results, we propose a simplified algorithm to calculate Kw from moderate PLDs. So this is an expression of the vascular signal, which can be simplified by taking logarithm on the both sides. So here, so you can see now the log delta MC signal is a very simple linear equation as a function of time or PLD, and the slope of the equation is only determined by Kw. So we can calculate Kw from voxel-wise linear regressions, and this algorithm is not sensitive to other parameters, such as arterial transit time. So here is a Kw map estimated from the previous developed DP Picasso technique. The resolution of the Kw map can be improved using the proposed new technique, and also the MCDW Picasso maps provides twofold more slices as compared to the DP Picasso method. And lastly, the new algorithm for Kw quantification does not require complicated modeling, which is also less sensitive to other physiological parameters. And in conclusion, we developed the MCDW Picasso technique with improved SNR and spatial resolution, and obtained comprehensive measurements of water kinetics using 3-compartment SPA modeling, and lastly developed algorithms for simultaneous KW and PSW mapping. Thanks so much for your attention. For more details, please come to the YIA session on Monday. Look forward to see you in Toronto.